Hey guys, this is Dr. L, and this is a short video lecture on microbial taxonomy, which is just a gigantic topic, and I'm going to just give you an overview of what is essentially a narrow slice of this topic, and we're going to focus on bacteria here. So taxonomy and nomenclature are two different things. Taxonomy is the study of how to classify or characterize what an organism is. So is it an animal? Within that, is it a vertebrate animal? Is it a dog? Etc., etc., right? And then nomenclature means to give a specific name. So one is to do the classifying and the other is to assign the name, okay? So nomenclature is an organismal name. And there are a lot of different ways to name organisms, and this is something that has evolved over time. So there are Latin or species names which fall into a system developed by Carl Linnaeus many years ago. Sometimes this is called binomial or trinomial nomenclature. Bi meaning two, tri meaning three. And so an example of that is the name given here, which is Salmonella enterica. And that is a particular type of bacterium. It's a gram-negative bacterium. And it is a common agent of foodborne illness and some other significant diseases. So that name there, Salmonella enterica, is a binomial Latin species name. Salmonella is capitalized, and it is the genus name, and enterica is not capitalized. It is the species adjective. And generally you should always italicize or underline these formal names. Within a species, you can have a subspecies, a strain, or a serovar. I know it's a lot of S terms, right? So a subspecies is the term, it's a very formal term, and it is a grouping, a taxonomic grouping below the level of a species. So this is used when you have a population of organisms here, microbes, and they have, a common and fairly significant distinct difference genetically from one another. So for example, within the Latin species or within the microbial species Salmonella enterica, there is a subspecies, and it gets shortened as SUBSP dot, another subspecies also called enterica. So within the species Salmonella enterica, there are actually at least 6 known subspecies. Enterica is one of those 6, and I'm mentioning it here because it is the most significant subspecies when it comes to human and other animal infections. So our genus is Salmonella, our species is enterica, and then our subspecies within that is again enterica. A serovar is a specific genetically distinct group of a particular species and or subspecies. I know, it's an and or an or. I told you this can be messy. And it is assigned on the basis of having a group of these organisms that cause their own unique immune reaction in the host. And this is because there are genetic differences, and they trigger the host's immune system a little bit differently. In other words, they have different antigenic properties. We'll be learning more about those terms later in this course. It's called a serovar because these organisms are identified on the basis of a human blood profile or chemistry, and blood profiles or chemistry are called serology. So this is all about how the host responds to the bacterium. So you really only see serovars assigned to organisms that are clinically relevant for that reason, because it's only in those organisms that cause disease that we are actually focusing on what the human blood chemistry would be. We don't care about it if it's something that doesn't cause disease. So here we see Salmonella enterica being the species name, subspecies enterica, and then within that there are many, many, many different serovars. One, for example, is called Serovar typhimurium. A strain is an additional level of categorization, and strains are a much less formal category. They're generally assigned by the lab that discovers the organism. They might be from a particular region or a particular patient if it's from a clinical lab, a particular strain, and there's probably some genetic differences between this strain and other strains, but it's not necessarily well established. So the strain identification often gives us a lot of information about where something came from and what sort of sample it came from. Like, did it come from urine, or did it come from a cluster of pregnant women in a particular geographic location, or something isolated from a particular location at a particular time. So for example, there is a particular strain called Salmonella enterica, subspecies enterica, known to be Cerevar tifimurium, and within that we can further cluster some of these organisms into strains, and there's a strain known as NCTC74. So all of this gets a little overwhelming, and it can help to have some visuals on it. So I'm not sure if you can see the very top of this. The axis of this says kingdom on the top, but this sort of goes from kingdom, phylum, class, order, family, genus, species, subspecies, Cerevar, and then I added in NCTC74, which was the strain ID. So the eubacteria are the bacteria. That's one of the 3 domains of life. I described that in another video. Within that, there are groups of bacteria, many different phyla of bacteria. One are called proteobacteria. Within the proteobacteria lie the gamma proteobacteria and others like alpha and beta. Within the gamma proteobacteria, there is an order called Enterobacteriales. Within those, there's something called a family called Enterobacteriaceae, and E. coli belongs to this as well. There's a specific genus called Salmonella. There are species within the Salmonella, and there are in fact more than 2, but we're focusing here just on Enterica. And within the species Enterica, there are these 6 subspecies, one of which is also called Enterica. And within the Enterica subspecies, there are 2 known Cerevars. There are actually more than that. This is an oversimplification, but there's a Cerevar called non-typhoidal and one called typhoidal. And within that, we've got the strain NCTC74. And I took this figure from a paper. The link is in the slideshow below. So on top of all of that, we have common names for organisms. So for example, a dog is Canis lupus familiaris. That's its Latin trinomial species name, but we just say dog for obvious reasons. Staph refers to organisms that belong to the genus Staphylococcus, but these kinds of infections from staph are so common that we just give them the nickname staph. MRSA means a particular type of Staphylococcus aureus, methicillin-resistant Staphylococcus aureus, but our common name is just the acronym MRSA. Strep bacteria are bacteria from a genus called Streptococcus, and that can be further divided, but they're such common pathogens that we just sort of collectively refer to them in common practice as strep. And there's a lot more to say about that, but I'm going to just mention that there are common names. Why should you actually care about any of this? This has tremendous relevance to healthcare. So you don't have to become the expert at this, but you do need to know that it matters and be able to look things up if you've got questions or you need to know more about a particular topic. So you cannot accurately diagnose the cause of infection if you cannot name the thing that is causing the infection. So in order to describe, work with, combat, detect, track, etc., any particular microbe, we really need to know some things about it. And in order to do that in a logical, sensible way, we have to be able to name it. So taxonomy and nomenclature matter. Antibiotic stewardship is critical in healthcare. We need to try to keep the antibiotics that we've got working to combat infections so we don't all die from like terrible bacterial infections as we did in the pre-antibiotic and pre-vaccine era. We really don't want to go back to those times. Using antibiotics safely and correctly is called antibiotic stewardship. And so knowing which particular organisms have antibiotic resistance genes and better being able to name and describe those is called antibiotic stewardship. And in order to do that correctly, you need to be able to name things correctly. You can't control things and study them, and that's infection control if you don't know what they are. Vaccine development depends on good systems of nomenclature and therefore taxonomy. We can't do personalized medicine if we don't know what is in a human's microbiome, or we don't know what the human is infected with, and what its genetic characteristics are, and that involves naming. We can't track strains if we don't know what they are called and we haven't got them classified. We can't understand new pathogens until we actually discover them, study them, and name them. That's all part of epidemiology. Understanding diversity in microbiome studies means doing a count or a census of how many different kinds of things make up your microbiome. And you can't count things if you can't name them and put them into categories. And then there's all sorts of basic research applications to this. So this is just a little slice of the pie. It focuses really on bacteria and not on viruses or fungi or other types of eukaryotes, but it's just a little bit of an introduction to some ideas around Latin species names and the ideas of strain, serovar, and subspecies, and a little bit about common names and why this should matter. So good luck with this.